The next session will be Keynote Lecture 8, RNA Therapeutics for Liver Cancers. This session will be chaired by Dr. Hiroyuki Isayama. This session is presented by Dr. Tushar Patel. Dr. Isayama, please.
Okay. Hello, everybody. I'm Dr. Isayama from Juntendo University. So this time, this keynote lecture, so I invited a very famous hepatologist from USA, Dr. Tushar Patel. Professor Patel, are you on the screen on this site? Okay, hello, hello.
Yes, I'm here.
Can you hear me?
Yes, I can hear you. Thank you.
Okay, we also hear your word very clearly. So today we will have this special lecture from the Professor Patel, and I want to introduce Professor Patel first. So Dr. Patel now professor in medicine, cancer biology, at Mayo Clinic, Florida, USA. So his areas of interest is liver and biliary cancers, and the clinical transplant hepatology, and the RNA, and the extracellular physical biology, and the therapeutics. And it is very important, emerging technologies and innovation in healthcare. So we are expecting a very exciting proposal, new therapy from Professor Patel. And he is currently the consultant in the Department of the Transplantation at the Mayo Clinic. And he was a dean for research at Mayo Clinic, Florida, 2014 and 22. And today's topic is a new therapy, the RNA therapeutics for liver cancer. Dr. Patel, are you ready?
Yes, thank you for the introduction. Will you be playing my presentation, or do you want me to give it live?
Video? Okay, please start.
Thank you for the introduction, and also thank you to the organizers for the kind invitation to present at this excellent meeting. I will review RNA therapeutics for liver cancers. This emerging area is garnering much attention these days, and I'll provide a very brief overview of the field and also try and highlight how these might be helpful and useful for liver cancers. I have no conflicts to declare. So, what are RNA therapeutics? So in short, these are medical treatments that use RNA molecules to target and treat disease. They can be used to regulate the expression of proteins for therapeutic effects. There are several different types of RNA therapeutics. These include messenger RNA, molecules that encode proteins to treat diseases, RNA interference molecules that block protein expression, microRNA and siRNA, antibody and oligonucleotide conjugates, and so on. These therapies are really in the early stages of development. However, they do have potential use for treatment of liver diseases and liver cancers. Nevertheless, there are many challenges that need to be overcome. First of all, the potential for using RNA for therapeutic purposes can really be best highlighted by the RNA-based vaccines that were introduced for COVID-19. These harness the body's immune system, and one form of the vaccine comprised of an RNA, which was enclosed within a lipid nanoparticle, in this case, towards a spike protein, and this was used to deliver the RNA into the body, where it generated an immune response, and it produced antibodies that targeted the viruses that harbor this protein. There are several different types of RNA that can be used for therapies. Each one of these has unique properties and potential applications. They include antisense oligonucleotides, messenger RNAs or mRNA, microRNAs, siRNA, and so on. siRNAs and aptamers are already in the clinic, and there is a robust pipeline in some therapeutic areas such as blood diseases and cancers. Of note, there are already several liver-directed RNA therapies that have been shown to be effective. So for example, mRNA therapies for protein replacement, such as enzyme replacement for metabolic disorders, RNA interference for hepatitis B, some very recent studies highlighting the efficacy of using this approach, and then siRNA-based therapies, including patisiran, the first one to be approved in 2018 to treat hereditary amyloidosis. There are several others that direct therapeutic RNA to the liver, and these have been used for various conditions such as acute hepatic porphyria, primary hyperoxaluria, polyneuropathy and amyloidosis, and even hyperlipidemia and atherosclerosis. So we've been exploring the use of RNA therapeutics for liver cancers by seeking to modulate gene expression, and our overall approach is summarized here. For delivery, a synthetic nanoparticle such as a lipid nanoparticle can be used, but so can extracellular vesicles that are biologically produced vesicles. The contents of these can include a payload of RNA therapeutics, such as siRNA or antisense oligonucleotides, or even proteins. And then finally, cell types targeting can be achieved by engineering these vesicles, for example, using surface antibodies or aptamers. So let me next review some of our clinical studies that demonstrate therapeutic efficacy of RNA-based therapeutics. In one study, we used antisense oligonucleotides to microRNA 221. These were loaded into milk-derived nanovesicles, and these nanovesicles, or MNV, they're easily isolated, they're non-toxic, and biodistribution studies have shown that they can accumulate quite quickly in the liver after systemic administration. So using intrahepatic human tumor cell xenografts in immunodeficient mice, we showed that MNVs loaded with antisense oligonucleotides resulted in tumor regression after the systemic administration. And moreover, they could be combined with nanovesicles loaded with doxorubicin for a synergistic effect. In other studies, we used a synthetic transgenic model of hepatocellular cancer, one induced by hydrodynamic injection of sleeping beauty transposase, the MET oncogene, and mutant beta-catenin. This is an immunocompetent model, and tumor formation occurs in 100% of the mice around after about 12 weeks. And for these studies, MNVs were loaded with siRNA to beta-catenin. They were administered over a 2-week period. And compared with controls, tumor burden was greatly reduced, and overall, there was a decrease in tumor burden and survival. In another study, we used RNA nanotechnology to design aptamers that recognize cell surface EpCAM, and then to incorporate these onto the surface of the MNVs that had been loaded with the therapeutic. Now, EpCAM is expressed on the cell surface of liver cancer and liver cancer stem cells. We then assess tumor uptake and the effects of these therapeutic targeted MNVs in vivo using subcutaneous xenografts of liver cancer stem cells and as well as Hep3B cells. Now, biodistribution studies, as shown here in panel A, showed high uptake in the stem cell xenograft tumors, and moreover, xenograft growth was greatly reduced with the use of these EpCAM targeted MNVs compared with controls. The specificity of uptake is shown here. So for these studies, EpCAM targeting MNVs were used to deliver siRNA to beta-catenin. In liver cancer stem cells, which have high expression of EpCAM, there's a marked reduction in beta-catenin expression. However, the effect is less prominent in HeLa cells, shown here on the right, that do not express EpCAM. These and several other preclinical studies that we have done in hepatocellular cancer, as well as cholangiocarcinoma, and also towards a variety of targets of interest, have demonstrated to us that RNA therapies can be useful for the treatment of liver cancers. However, despite these compelling examples, there's still many challenges that need to be overcome. One challenge is specificity. We need to ensure that RNA therapeutics are specific enough to target the desired protein without affecting other proteins in the body. Another one is delivery. Delivering the RNA molecules to the right target cells in the body is an important need that needs to be optimized for each indication. Yet another challenge is stability. RNA molecules, they're quite sensitive to degradation, and this can limit the effectiveness of therapeutics. The therapeutics can be toxic. We do need to make them safe for use in the body. And then I should also mention that therapeutics, RNA therapeutics, like any others, are subject to strict regulatory oversight, and ensuring compliance with regulatory requirements can sometimes be challenging. So overall, these are challenges, but advances in technology, advances in research are helping us to overcome some of these and bring RNA therapeutics closer to clinical use. So how might we approach these challenges? So obviously having the right target, one that's specific for the disease, is necessary. We also need to develop methods for targeting RNA molecules and to ensure that the delivery to the right target cells occurs, as well as to ensure that the stability is being maintained. Now, these can be addressed by using an appropriate carrier and in some cases, appropriate targeting approaches. Some other important aspects are tolerability and toxicity of the RNA. Here, we can use RNA modifications to achieve the desired effects, particularly for messenger RNAs. And then finally, we need permissive regulations that span the spectrum from drug approval to standards for potency in manufacturing to acceptable conduct of clinical trials. There are several different carrier systems that can be used to deliver RNA therapeutics to target cells in the body. I've outlined several studies already that used vesicles or biological nanovesicles. However, there are several other types of carrier systems as illustrated here. And these include liposomes, which are tiny spherical structures made of lipids. They also include polymeric nanoparticles, which are tiny particles that are made of polymers. Other delivery vectors are the use of viruses that have been genetically modified to carry the RNA molecules into cells. Now, obviously, the most appropriate carrier system will depend on the specific RNA therapeutic that's being tested, as well as the specific target cells in the body. So next, let's consider how RNA, such as messenger RNA, could be modified to generate a useful therapeutic. Now, mRNA is immunogenic. It'll trigger an innate immune sensing response and result in the production of inflammatory cytokines. Now, this could be helpful for vaccines, but it's not really necessary and is a hindrance for if you're using mRNA for protein replacement or for regenerative applications. However, fortunately, RNA can be modified in many, many different ways. And in fact, natural RNA contains hundreds of modifications. We know that some viruses and bacteria will in fact modify their genetic material to be able to evade immune recognition. So here are some common approaches that we can use. These include modifications of the 5' cap to increase the translational efficiency, to increase the rates of translation initiation. They may also decrease some repression of translation that can occur that's triggered by the immune responses. Other approaches include modifications in the poly-A tail, and these can increase resistance to exonucleases. Finally, we can also have modifications to nucleotides themselves. For example, one common approach is replacing uridine with pseudouridine or its methyl analog. And then finally, we can also consider the use of synthetic oligos. These allow us to increase the diversity, and they can also be helpful in designing messenger RNAs with an extended lifespan. So the next consideration is the pathway to development of treatments. Going beyond just designing the RNA for specific targets and showing the efficacy in preclinical models, we need to consider the process of drug development, including synthesis, preclinical testing, cGMP production towards clinical testing. And streamlining the processes at each step can be necessary, will help to accelerate the development and then the eventual approval for use in the clinic. So in closing, there's several opportunities to accelerate the development of RNA therapeutics. These have tremendous potential for the treatment of liver cancers. These opportunities include advances in technology, such as new delivery systems, new methods of RNA synthesis that can make it faster and easier to develop new therapeutics. They can involve collaborations and partnerships, for example, between researchers, pharmaceutical companies, and regulatory agencies, which will all be necessary to accelerate the development of therapeutics by sharing expertise, sharing resources, and then also by increasing funding to support the development not only of new technologies, but also of being able to translate the research findings into clinical applications. And by taking advantage of these, we will hopefully bring RNA therapeutics closer to clinical use for the treatment of liver cancers in the future. And I will just end with that and once again offer my thanks to the organizers for allowing me to participate in this very excellent meeting. Thank you.
Professor Patel, thank you very much for excellent talk about new therapy using the RNA. So we know the RNA therapy is very effective for the COVID-19 vaccination. So, but I'm not familiar with the RNA therapy for the malignancies. So, firstly, please tell us, is there any therapy for the other areas malignancy? So the already in a clinical use or in a very near step. Please tell us the situation.
Thank you for your question. So the clinical use of RNA therapeutics is really accelerating now with many applications in the liver. I think it's just a matter of time before we see specific targets for oncological targets. I should mention that the use of RNA therapeutics for liver cancers is not new. In fact, probably more than 12, 14 years ago, there were descriptions of RNAi targeting VEGF and KSP for liver cancers with preclinical studies showing some very dramatic effects. But the challenge has been, how do you take those proof of concept studies into an effective therapeutic? And so the pathway is quite long. You have to overcome some of the issues I mentioned, such as delivery to the appropriate cells, but also making sure that they're safe and effectively delivered. But I think that many of these challenges have been overcome with some of the new therapies over the last 3 or 4 years. We now have several approved drugs, which are all targeting the liver, as a matter of fact, because it's very easy to target the nanoparticles to the liver. And so I think it's just a matter of time before we start seeing these now in the oncological area, and then eventually in liver cancers.
Thank you very much. A question from the audience, please.
Thank you very much for your very informative talk. My name is Charupong Sangwinmi from Thailand. So I have a question about cholangiocarcinoma, because you said that for targeting the liver is quite easy, but for cholangiocarcinoma, I think it's very high. There's more plastic. There are a lot of fibrosis around the cancer cells. Do you think the RNA therapy going to be useful for this kind of cancer?
Yes. As a matter of fact, we do have some preclinical data in which we have directed the vesicles that are delivering the RNA therapeutic to cholangiocarcinoma cells. Mostly the strategy that we've used for this is to target EpCAM. EpCAM is found in highly expressed on the surface of many epithelial cancers, including cholangiocarcinomas. And we've been able to show that we can very effectively deliver RNA therapies by designing vesicles that will recognize cell surface EpCAM on cholangiocarcinoma cells. So this work is going to be submitted soon, and but so there's a pathway. We are also looking at other cholangiocarcinoma specific markers and cell specific markers to use for targeting. So the key here is actually being able to direct the vesicles to the cell of interest and finding a cell specific marker that can be robust enough to be able to encourage the uptake of these vesicles.
Okay. I have another question about the administration of the drug or the vesicle that you have developed. So which is the best route to apply this drug, by intravenous injection, or we can just use like the intramuscular injection like the RNA vaccine?
Yes, there are several different approaches that can be taken, and it really depends on the type of RNA molecule that you're using. So, for example, if you're using just plain messenger RNAs that's modified, or whether you're using sRNAs, some of the therapeutics that are currently in the clinic don't use vesicles. They are sRNA that are conjugated with, again, a cell-specific recognition molecule. And so those could be amenable to direct injection. Others might require the administration of vesicles. the biological vesicles that we have used can also be delivered orally. So the bottom line is that there could be a variety of different approaches, oral, subcutaneous, intravenous, or direct intratumoral, depending again on the therapeutic, the delivery approach that's taken, and what your goals are.
Thank you very much.
And other delivery route is we have a transarterial, also injection to the tumor directly. So it is more efficient. How do you think about these ideas?
I think that any way that you can increase the effective therapeutic at the target site, in this case, the liver tumors, will obviously, I think, have the potential of increasing the efficacy. With many of the synthetic nanoparticles that are used as carriers for the therapeutics, there's very high uptake in the liver, particularly with the macrophages, Kupffer cells. And so there's a higher local concentration of the liver. But I think with regards to the treatment of liver tumors, it's very, very attractive to be able to directly infuse these through the hepatic artery.
Okay, thank you very much. Is there any question or comment from audience? Okay, Dr. Patel, thank you very much for very interesting your study. Thank you
very much for the opportunity.
I want to close this keynote lecture.
Thank you very much, chairs and lectures. The next session will start at 12:30.